A person with locked-in syndrome is fully conscious and aware, but completely unable to move or speak, typically retaining only vertical eye movements or blinking to communicate. This clearly differentiates it from a vegetative state, where the patient may have wakefulness, eyes open, sleep-wake cycles, but lacks conscious awareness of self or environment. Its core distinction: a patient with locked-in syndrome, the mind is intact while the body disconnected. While a vegetative state, the body may appear active, but the mind is not consciously engaged. It's like being in prison in a body you have no control. Locked-in syndrome is most commonly caused by a blockage of the basilar artery that cuts off blood flow to the brainstem region that contains the motor pathways responsible for voluntary movement. Immediate functional state: the brain remains fully active, with cortical neurons firing in stable, coordinated patterns that preserve awareness, language formation, memory recall, and continuous perception of the environment. Motor commands are still generated in the motor cortex with precision, structured as organized electrical signals intended to move the limbs, control the face, and produce speech. These commands form correctly and repeatedly, reflecting intact cognitive function and preserved intention. Yet none of these signals produce movement once they begin descending toward the brainstem. This creates a complete separation between intention and execution, while consciousness remains intact. Motor signals travel downward through established neural pathways without distortion, maintaining their timing and structural integrity as they leave the cortex and enter deeper brain structures. They pass through regions responsible for coordinating voluntary movement, carrying detailed instructions for muscle activation that would normally produce immediate response. At a specific point within the brainstem, this transmission abruptly stops, preventing activation of lower motor neurons that control skeletal muscle. The interruption is absolute, not partial, and no degraded signal reaches the muscles. The body remains entirely unresponsive, despite continuous command generation from the brain. Skeletal muscles remain physiologically capable of contraction, with intact neuromuscular junctions and functional peripheral nerves that are ready to respond to incoming signals. However, no effective command reaches them, resulting in complete absence of voluntary movement, no adjustment of posture, and no ability to initiate speech or facial expression. Internally, the brain continues issuing commands in repeated attempts to produce movement, reinforcing the persistence of intact motor intention. There is no delay or fluctuation in this failure, only consistent absence of output. The body exists in a state of readiness without activation. Automatic functions remain preserved because they are controlled by brainstem systems that operate independently of voluntary pathways. Breathing continues through autonomic respiratory centers, maintaining oxygen exchange without requiring conscious input. Heart rate, basic reflexes, and essential survival functions remain stable, indicating that the failure is not global but highly selective. This distinction reveals that the disruption specifically targets voluntary motor pathways rather than the entire nervous system. The body continues to sustain life while losing the ability to act. Visual processing remains intact, with the eyes open and capable of receiving and interpreting light, motion, and spatial information. The visual cortex continues functioning normally, allowing full awareness of the surrounding environment. Vertical eye movement and blinking are preserved because the neural pathways controlling these functions remain unaffected by the disruption. This creates a limited but critical channel for interaction with the environment. Even within widespread paralysis, selective preservation defines the condition. Horizontal eye movement is impaired due to involvement of specific brainstem structures responsible for coordinating lateral gaze. This restriction further narrows the already limited capacity for communication and environmental interaction. The contrast between preserved vertical movement and impaired horizontal movement reflects precise anatomical selectivity rather than generalized damage. Each preserved or lost function corresponds directly to specific pathway involvement. The pattern is structured, not random. All conventional forms of communication collapse simultaneously, including speech, gestures, writing, and facial signaling. The inability to activate muscles responsible for expression removes all outward signs of cognition. Only minimal eye-based communication remains, dependent on preserved vertical gaze and blinking. This creates a single narrow output channel connecting an intact cognitive system to the external world. Communication becomes constrained to the smallest surviving pathway. Cognitive function remains fully intact, with preserved reasoning, perception, and internal language. The individual continues to understand surroundings, process conversations, and form responses internally. There is no impairment in awareness, comprehension, or thought generation. The failure exists entirely at the level of execution, not cognition. This produces a state where the mind remains active while the body remains silent. Auditory input is processed normally, with sound signals reaching the auditory cortex and being interpreted in real time. Spoken language is understood, questions are processed, and meaning is fully constructed internally. Despite this, no verbal response can be produced due to the loss of motor control over speech mechanisms. The inability to respond does not reflect lack of understanding, but lack of execution. The separation between input and output becomes absolute. Sensory pathways remain functional, allowing pain, touch, and other stimuli to be consciously perceived. Signals from the body reach the brain without disruption, maintaining full sensory awareness. However, the absence of motor output prevents any visible reaction to these stimuli. The body can feel, but cannot respond. This reinforces the dissociation between perception and action. Time perception remains stable, allowing continuous awareness of events and the ability to track duration. Memory formation continues, and the sequence of experiences is preserved without interruption. There is no distortion in temporal awareness, despite the absence of movement. The internal sense of continuity remains intact. The body remains still while time continues normally. Sleep and wake cycles persist under normal neurological control, allowing transitions between states of consciousness. The brain maintains its regulatory rhythms, shifting between alertness and rest as it would under normal conditions. Despite these changes in state, the absence of voluntary movement remains constant. The condition does not fluctuate with consciousness level. The paralysis is stable across all states. This condition is identified early as locked-in syndrome, defined by preserved consciousness combined with near total loss of voluntary motor function due to disruption of descending motor pathways. The defining feature is not loss of awareness, but loss of execution. The brain remains capable of generating complete behavioral intention. The body cannot carry it out. This establishes the fundamental identity of the condition. The disconnection is highly selective, affecting motor execution systems while leaving higher cognitive functions intact. This creates a precise neurological state where intention, awareness, and perception continue without any ability to act on them. Each preserved function highlights the specificity of the disruption. Each lost function reflects pathway-level failure rather than global damage. The system is partially intact but functionally disconnected. The environment remains fully perceptible, including visual detail, auditory input, and spatial awareness. The individual remains aware of surroundings in real time, with no loss of external perception. Interaction is limited to minimal eye movement, restricting communication to a narrow channel. The body cannot engage with the environment despite full awareness of it. Awareness persists without expression. Repeated attempts to initiate movement continue within the brain, producing consistent and correctly structured motor commands. These commands follow normal neural pathways until they reach the point of interruption, where they fail completely. Each attempt produces the same result, reinforcing the stability of the failure. There is no adaptation or compensation that restores movement. The system remains locked in a state of continuous intention without execution. Pathway disconnection, motor output failure. Motor commands originate in the primary motor cortex, where organized electrical patterns encode direction, force, and coordination for voluntary movement. These signals are transmitted downward through the corona radiata and converge into the internal capsule, a compact structure that funnels nearly all descending motor information into a confined anatomical space. From here, the signals enter the brainstem as tightly bundled fibers, maintaining precision and timing as they travel. There is no degradation in signal quality at this stage. The command remains complete, structured, and ready for execution. Within the brainstem, these descending signals form two critical pathways, the corticospinal tract and the corticobulbar tract. The corticospinal tract carries motor commands destined for the limbs and trunk, transmitting instructions from the cortex toward spinal motor neurons. The corticobulbar tract carries commands toward cranial nerve nuclei, controlling facial expression, speech, chewing, and swallowing. Both tracts are densely packed together within the ventral portion of the brainstem, creating a region where multiple motor systems are anatomically concentrated. This convergence creates efficiency under normal conditions. It also creates vulnerability. The ventral brainstem, particularly the region known as the Pons, becomes the critical site where these pathways can be interrupted simultaneously. In this confined area, the corticospinal and corticobulbar tracts run in close proximity with minimal redundancy or alternate routing. A single lesion in this location can block nearly all descending voluntary motor signals at once. The interruption does not weaken the signal gradually. It eliminates transmission entirely. Once this interruption occurs, motor commands generated in the cortex can no longer reach lower motor neurons in the spinal cord or cranial nerve nuclei. The muscles themselves remain structurally intact and physiologically capable of contraction. Peripheral nerves remain functional and ready to transmit signals. Neuromuscular junctions remain responsive. However, without descending input, none of these systems activate. This creates a state where the body is capable of movement at every peripheral level, but receives no instruction from the central command system. The failure is not located in the muscle, the nerve, or the neuromuscular junction. It is located in the transmission pathway that connects intention to execution. The system is intact but disconnected. The result is complete motor silence. Cranial nerve functions, dependent on corticobulbar input, are equally affected by this disconnection. Muscles responsible for speech cannot be activated, preventing phonation despite intact vocal structures. Facial muscles receive no voluntary command, eliminating expression even when emotional processing remains active. Swallowing becomes impaired because coordinated activation of multiple muscle groups cannot be initiated. These losses occur simultaneously because they share the same descending pathway. Limb movement is also completely absent because spinal motor neurons receive no descending input from the corticospinal tract. Without this input, muscles of the arms, legs, and trunk cannot initiate contraction. There is no partial movement or weakened response. There is only absence of activation. The body remains motionless despite preserved structural capability. Postural control is lost for the same reason, as muscles responsible for maintaining upright position cannot be voluntarily engaged. Balance systems may remain intact at a sensory level, but they cannot translate into corrective movement. The body becomes dependent on external positioning. Voluntary adjustments are no longer possible. Stability becomes passive rather than active. Voluntary control of breathing is also affected, as descending input to respiratory muscles is interrupted. However, automatic breathing continues because it is regulated by brainstem centers that function independently of voluntary pathways. This distinction highlights the specificity of the disruption. The system does not lose all function. It loses only the ability to voluntarily control certain functions. The preservation of vertical eye movement reveals an important anatomical exception. Neural pathways controlling vertical gaze are located in regions of the brainstem that remain unaffected by the lesion. This allows upward and downward eye movement to persist even when other motor pathways are interrupted. It becomes the primary remaining channel for voluntary output. Communication depends on this preserved function. In contrast, horizontal eye movement is impaired due to involvement of structures responsible for lateral gaze coordination within the brainstem. This further restricts communication and demonstrates how small differences in anatomical location determine functional survival. Not all motor systems are equally vulnerable. Their fate depends on their pathway location relative to the lesion. The disconnection created by this pathway interruption is precise and consistent. Every motor command generated in the cortex follows the same route and encounters the same point of failure. There is no variability in outcome. Each signal is blocked at the same level. This creates a stable and predictable pattern of complete motor output failure. Internally, the brain continues to generate commands based on intention and sensory input. These commands are produced repeatedly, attempting to initiate movement in response to the environment. However, no feedback is received indicating successful execution. The system operates without confirmation of action. This creates a loop of continuous command generation without response. The absence of feedback does not correct the system because the brain is not receiving signals indicating pathway disruption. It continues to operate under the assumption that commands should produce movement. Each attempt reinforces the failure. The disconnect remains uncorrected. The pattern becomes continuous. This is not paralysis caused by muscle damage or nerve degeneration. It is paralysis caused by interruption of communication between intact systems. The muscles are capable. The nerves are functional. The brain is active. The connection between them is severed. This defines the condition at its core. The body remains structurally able to move, but functionally silent because the pathways that translate intention into action no longer transmit any signal. The failure is centralized, precise, and complete. There is no alternate route that compensates for the loss. The system remains locked in a state of continuous intention without execution. Brainstem localization, ventral pons. The point of failure is localized within the brainstem, specifically in the ventral region of the pons, where descending motor pathways are densely concentrated into a compact anatomical structure. This region acts as a critical transit zone for nearly all voluntary motor signals traveling from the cortex to the body. Its compact design allows efficient signal transmission under normal conditions. It also creates a single point of failure when disrupted. Damage here does not spread gradually across systems, but instead interrupts multiple pathways at once. The pons sits between the midbrain and the medulla, forming part of the brainstem that connects higher cortical function to spinal and peripheral systems. Within its ventral portion lie tightly packed fiber tracts, including the corticospinal and corticobulbar pathways responsible for voluntary movement. These fibers are arranged in parallel bundles with minimal separation, allowing a localized lesion to affect them simultaneously. There is little redundancy within this structure. Once damaged, alternate routing is not available. The ventral pons is specifically vulnerable because it contains the descending motor tracts without containing the primary centers responsible for consciousness. The reticular activating system, which maintains wakefulness and awareness, is located in adjacent but distinct regions of the brainstem that are often spared. This anatomical separation explains why consciousness remains intact despite complete motor paralysis. Awareness continues because the systems that sustain it are not directly affected. The failure is selective. Within this confined region, the lesion disrupts signal transmission at the level where upper motor neurons would normally connect to lower motor pathways. This interruption prevents motor signals from progressing further downward into the spinal cord and cranial nerve nuclei. The effect is immediate and complete. No degraded signal passes through. The connection is effectively severed at a single anatomical point. The density of fiber tracts in the ventral pons means that even a small lesion can have widespread functional consequences. A disruption measuring only millimeters can eliminate the output of entire motor systems. This scale mismatch between lesion size and functional impact defines the severity of the condition. The damage is localized, but the effect is global in terms of motor function. The body loses control because the pathways are concentrated, not distributed. The dorsal portion of the pons, which contains structures related to sensory processing and coordination, may remain relatively preserved depending on the extent of the lesion. This contributes to the preservation of sensory awareness and certain reflexes. The contrast between dorsal preservation and ventral destruction reinforces the anatomical specificity of the condition. Different regions of the same structure produce entirely different outcomes. Localization determines function. Cranial nerve nuclei located within or near the pons may retain partial function if they are not directly involved in the lesion. However, without corticobulbar input, voluntary control over these nuclei is lost. Reflexive or automatic responses may still occur, but intentional activation is absent. This distinction further emphasizes the separation between voluntary and involuntary systems. The pathways are present, but the command signal is missing. The preservation of vertical eye movement is explained by the location of gaze centers in the midbrain rather than the pons. These centers remain functional because they lie outside the primary area of damage. This allows upward and downward eye movement to persist even when most other motor pathways are interrupted. It becomes a critical functional remnant of a largely disconnected system. Its survival reflects precise anatomical boundaries. Horizontal gaze centers, however, are located within the pons and are often affected by lesions in this region. This results in impaired lateral eye movement, further limiting communication and interaction with the environment. The difference between vertical and horizontal gaze function highlights the importance of exact lesion location. Small shifts in anatomical involvement produce distinct functional outcomes. The pattern is consistent with structural organization. The ventral pons also contains pathways involved in coordination between the cortex and cerebellum, contributing to fine motor control under normal conditions. When disrupted, these pathways no longer transmit corrective feedback or coordination signals. However, because voluntary movement is already eliminated, the loss of coordination pathways becomes secondary. The primary failure remains the interruption of motor output. Coordination cannot occur without movement. Blood supply to this region is primarily provided by branches of the basilar artery, making it particularly vulnerable to vascular events that can rapidly compromise oxygen delivery. When this supply is interrupted, the affected tissue undergoes ischemia, leading to rapid functional loss. The compact structure of the ventral pons means that vascular disruption has immediate and widespread consequences. The system fails quickly once blood flow is compromised. The localization of the lesion defines the condition completely, as it determines which pathways are interrupted and which are preserved. The ventral pons represents a convergence point where multiple critical motor systems intersect. Damage here produces a predictable pattern of preserved consciousness with complete motor paralysis. This pattern is not random. It is anatomically determined. This region functions as a bottleneck in the nervous system, where large volumes of motor information pass through a narrow space. Under normal conditions, this design supports efficient communication between the brain and the body. Under pathological conditions, it becomes a vulnerability. A single disruption can block all downstream communication. The system depends on the integrity of this narrow passage. The result of this localization is a condition where the brain remains capable of generating intention and awareness while the body is unable to execute any voluntary action. The anatomical separation between preserved and destroyed systems creates the defining characteristics of the syndrome. The failure is not distributed across the brain. It is concentrated within a specific structural region. This concentration explains both the severity and the precision of the outcome. Vascular event. Basilar artery occlusion. The initiating event occurs within the basilar artery, the primary vessel responsible for delivering oxygenated blood to the brainstem, including the ventral pons, where critical motor pathways are concentrated. Blood normally flows upward through this artery in a continuous, pressurized column, supplying small penetrating branches that feed the dense neural tissue within the pons. This flow must remain uninterrupted to sustain neuronal function. Even brief reductions in perfusion begin to affect energy-dependent processes. When flow is blocked, the system fails rapidly. A thrombus forms within the vessel, often developing over areas of endothelial damage caused by chronic vascular stress, such as hypertension or atherosclerotic plaque formation. Platelets adhere to the damaged surface, initiating a cascade of clot formation that gradually enlarges within the arterial lumen. As the clot grows, it narrows the channel through which blood flows, increasing resistance and disrupting normal circulation. Turbulence develops around the obstruction, further promoting clot stability. The vessel transitions from partial flow to critical narrowing. Once the thrombus reaches a size sufficient to obstruct the artery, blood flow beyond the point of occlusion drops sharply or stops entirely. The penetrating branches that supply the ventral pons are immediately affected, losing their source of oxygen and glucose. Neurons within this region are highly dependent on continuous energy supply and have minimal tolerance for interruption. Within seconds, metabolic processes begin to fail. Electrical activity becomes unstable as energy reserves decline. Oxygen deprivation initiates a cascade of ischemic events at the cellular level, beginning with failure of oxidative phosphorylation within neuronal mitochondria. ATP production drops rapidly, removing the energy required to maintain ion gradients across the cell membrane. Sodium and calcium begin accumulating neurons, while potassium leaks out, disrupting membrane potential. This ionic imbalance alters electrical stability and prepares the cell for structural failure. The neuron shifts from controlled signaling to metabolic crisis. As ischemia continues, the lack of ATP disables ion pumps that normally regulate intracellular and extracellular ion distribution. Calcium influx becomes uncontrolled, activating enzymatic pathways that damage cellular structures, including proteins, membranes, and DNA. Glutamate, an excitatory neurotransmitter, is released in excessive amounts, further amplifying calcium entry into neighboring neurons. This process, known as excitotoxicity, spreads damage beyond the initial site of ischemia. The injury expands within the affected region. The ventral pons, due to its dense concentration of motor pathways, experiences simultaneous disruption across multiple fiber tracts as ischemia progresses. The corticospinal and corticobulbar pathways lose their ability to transmit signals, not because they are mechanically severed, but because the neurons and axons that sustain them can no longer maintain functional integrity. Signal transmission collapses as cellular structures degrade. The interruption becomes complete. Motor output ceases. The transition from ischemia to infarction marks the point where tissue damage becomes irreversible. Cells that have lost membrane integrity, mitochondrial function, and structural stability cannot recover even if blood flow is restored. Necrosis begins within the core of the affected region, where energy depletion is most severe. Surrounding areas may experience partial damage, but the central zone undergoes permanent destruction. This defines the biological boundary between reversible and irreversible injury. Timing plays a critical role in this process, as the duration of blood flow interruption determines the extent of tissue loss. Within minutes, significant neuronal damage occurs in energy-dependent regions like the brainstem. Unlike other tissues, neurons have limited capacity for anaerobic metabolism and cannot sustain prolonged periods without oxygen. The progression from functional impairment to structural death is rapid. Delay in restoration of blood flow increases the volume of irreversible damage. The occlusion of the basilar artery does not produce Gradual symptom onset, but often leads to sudden and catastrophic failure of motor pathways. Because the ventral pons is affected as a single functional unit, the resulting deficits appear simultaneously rather than sequentially. There is no staged progression of weakness. There is immediate and complete loss of voluntary movement once the pathways are compromised. The onset reflects the anatomical organization of the region. Collateral circulation in the brainstem is limited, providing minimal alternative blood supply when the primary artery is blocked. This lack of redundancy increases vulnerability to ischemic injury. Unlike other regions of the brain that may receive partial compensation from neighboring vessels, the ventral pons relies heavily on direct branches from the basilar artery. When this supply is lost, compensation is insufficient. The tissue becomes dependent on restoration of primary flow. Reperfusion, if achieved early, may restore function in partially affected areas where cells have not yet reached irreversible damage. However, in cases where infarction has already occurred, restoring blood flow does not reverse the loss of destroyed neurons. The pathways remain permanently disrupted. The distinction between salvageable tissue and infarcted core determines clinical outcome. Once necrosis is established, recovery is limited. The vascular event defines the origin of the condition, linking systemic vascular pathology to localized neurological failure. The clot itself is small relative to the overall circulatory system, yet its position within a critical artery amplifies its impact. A single obstruction interrupts the flow to a densely packed region responsible for essential motor functions. The scale of the effect is determined by location, not size. This reflects the hierarchical structure of the nervous system. The result of basilar artery occlusion is a rapid transition from normal neural function to complete disruption of voluntary motor pathways, while consciousness remains preserved due to the sparing of adjacent structures. The vascular event sets the entire cascade in motion, transforming a localized blockage into a global loss of motor output. The mechanism is direct, anatomical, and immediate. Blood flow stops, neurons fail, and communication pathways collapse. Cellular death, infarction, and necrosis. The loss of blood flow to the ventral pons immediately disrupts cellular metabolism, forcing neurons into an energy crisis that begins within seconds of ischemia. Oxygen delivery stops, and oxidative phosphorylation inside mitochondria can no longer produce ATP, the molecule required to sustain all active cellular processes. Without ATP, neurons cannot maintain the ion gradients that stabilize their membrane potential. Electrical signaling becomes unstable and progressively fails. The cell shifts from controlled function to metabolic collapse. As ATP levels fall, membrane ion pumps such as the sodium-potassium ATPases cease functioning, allowing sodium and calcium ions to accumulate the neuron while potassium leaks out into the extracellular space. This ionic imbalance causes depolarization of the cell membrane, triggering uncontrolled electrical activity that further disrupts normal signaling. The neuron can no longer distinguish between activation and rest. This loss of regulation marks the transition from functional impairment to structural vulnerability. The cell is no longer operating within physiological limits. Calcium influx becomes the central driver of cellular damage, as excessive intracellular calcium activates enzymes that degrade proteins, lipids, and nucleic acids within the neuron. These enzymes begin breaking down the cytoskeleton, destabilizing the cell membrane and damaging internal organelles. Mitochondria, already impaired by lack of oxygen, become further compromised by calcium overload, reducing any remaining capacity for energy production. The cell enters a self-destructive state driven by biochemical imbalance. Structural integrity begins to fail. Excitotoxicity amplifies this damage as glutamate is released in excessive amounts into the synaptic environment due to membrane instability and impaired reuptake mechanisms. Neighboring neurons are exposed to this elevated glutamate concentration, leading to further calcium influx and propagation of injury across adjacent cells. The damage is no longer confined to the initial site of ischemia. It spreads to connected neural networks within the affected region. The injury expands beyond the original vascular territory. As these processes continue, the neuron loses its ability to maintain membrane integrity, leading to swelling, rupture, and eventual cell death. This form of cell death, known as necrosis, is characterized by uncontrolled breakdown of cellular components and release of photic, intracellular contents into the surrounding tissue. The local environment becomes inflammatory, attracting immune cells that contribute to further tissue damage. The area of infarction becomes structurally and functionally destroyed. Recovery of these cells is no longer possible. Within the ventral pons, this cellular death affects large numbers of axons within the corticospinal and corticobulbar tracts simultaneously. These axons rely on intact neuronal cell bodies and continuous metabolic support to maintain signal conduction. Once the supporting neurons undergo necrosis, the axons lose their functional integrity. Signal transmission through these pathways ceases completely. The interruption becomes permanent at the structural level. The progression from ischemia to infarction defines a critical biological threshold where functional impairment becomes irreversible damage. Early in ischemia, some neurons may remain viable if blood flow is restored quickly, allowing partial recovery of function. However, once necrosis occurs, the affected tissue cannot regenerate. The central nervous system has limited capacity for neuronal replacement. Lost neurons are not restored. The pathways they supported remain permanently disrupted. Axonal degeneration follows neuronal death as disconnected axons begin to break down in a process that removes non-functional neural connections. This further reinforces the loss of communication between the cortex and the body. Even if upstream structures remain intact, the absence of viable pathways prevents signal transmission. The system cannot bypass the damaged region. The disconnection becomes anatomically fixed. Inflammatory responses within the infarcted area contribute to secondary injury as immune cells release cytokines and other mediators that can damage surrounding tissue. Edema may develop, increasing pressure within the confined space of the brainstem and potentially affecting adjacent structures. While this secondary process can expand the area of damage, the primary loss of motor pathways has already occurred. The critical functions are already compromised. The system has passed the point of recovery. The compact structure of the ventral pons means that even a small infarct can destroy a high density of critical fibers. This concentration amplifies the functional impact of cellular death. A limited volume of tissue loss produces extensive neurological consequences. The severity of the condition is determined not by the size of the infarct, but by its location within a critical pathway convergence zone. This reflects the organization of the brainstem. The reticular activating system, located in adjacent regions, may remain structurally intact despite the infarction. This allows consciousness to persist even as motor pathways are destroyed. The preservation of awareness alongside loss of movement is a direct result of selective cellular death within specific anatomical regions. The pattern of damage determines the clinical presentation. It is not random but anatomically defined. At this stage, the system has transitioned from disrupted function to permanent structural loss. Neurons have died, axons have degenerated, and pathways have been eliminated. There is no mechanism within the central nervous system to restore these exact connections. Compensation is limited because alternate pathways do not exist for these functions. The disconnection remains fixed. The infarcted region becomes a non-functional segment within the neural network, interrupting communication between intact upstream and downstream systems. The brain continues to generate motor commands, but these commands encounter a region where no viable pathways exist. The signal cannot pass through dead tissue. The failure is complete and irreversible. This cellular death defines the permanence of the condition, establishing a biological boundary beyond which recovery of voluntary motor function is not possible. The damage is not temporary suppression or reversible dysfunction. It is structural elimination of the pathways required for movement. The system has lost the components necessary for execution. Irreversibility boundary. The transition from functional disruption to permanent loss occurs at the moment when neuronal damage crosses the threshold from metabolic failure into structural destruction. During early ischemia, some neurons remain viable if oxygen delivery is restored quickly, allowing ion gradients and cellular processes to recover. This window is narrow and dependent on time, temperature, and local perfusion conditions. Once ATP depletion persists long enough to trigger membrane breakdown and enzymatic degradation, recovery is no longer possible. The system moves from reversible dysfunction into irreversible injury. Within the ventral pons, this boundary is defined by the loss of structural integrity in neurons and axons that form the corticospinal and corticobulbar tracts. These pathways rely on continuous cellular support to maintain signal conduction, and once the supporting neurons undergo necrosis, the axonal framework collapses. The damage is not a temporary interruption of signaling, but a physical loss of the structures required for transmission. There is no residual pathway that can carry the signal around the lesion. The communication line is permanently severed. Axonal regeneration within the central nervous system is extremely limited, particularly in highly specialized and densely organized regions such as the brainstem. Unlike peripheral nerves, central axons lack the biological environment necessary for effective regrowth, including supportive scaffolding and growth-promoting signals. In addition, inhibitory molecules released after injury prevent new connections from forming across the damaged area. Even if partial regrowth occurs, it cannot reconstruct the precise organization required for coordinated motor output. Functional restoration does not occur. The concept of irreversibility in this condition is not based on duration alone, but on structural outcome. A brief interruption that resolves before cellular breakdown may allow recovery, while a slightly longer interruption that triggers necrosis results in permanent loss. This creates a sharp biological boundary rather than a gradual decline. The system does not slowly degrade into paralysis. It crosses a threshold beyond which recovery is no longer biologically possible. Once this boundary is crossed, the brain continues to generate motor commands that encounter a region of non-functional tissue. These signals cannot pass through the infarcted area, and no alternate pathway exists to reroute them. The upstream systems remain active and downstream muscles remain capable, but the connection between them has been eliminated. This produces a stable state where intention persists without execution. The failure becomes fixed. Neuroplasticity, the brain's ability to reorganize connections, has limited capacity in this context because the lost pathways are highly specific and anatomically constrained. While some degree of adaptation may occur in surrounding regions, it cannot replace the direct descending control required for voluntary movement. The functions carried by these tracts cannot be reassigned to distant areas without the necessary structural connections. Plasticity cannot bridge a complete anatomical disconnection. The limitation is structural, not functional. The preservation of consciousness reinforces the perception of irreversibility because awareness continues to generate intent without any change in outcome. Each attempt to move produces the same absence of response, confirming that the system has reached a stable endpoint. There is no fluctuation or gradual improvement when structural loss is established. The condition remains consistent over time. The pattern is fixed by anatomy. In clinical terms, this boundary defines prognosis, as recovery of voluntary motor function depends on whether any viable neural tissue remains within the affected pathways. If the infarction has fully destroyed the ventral pons motor tracts, functional recovery is not expected. Partial lesions may allow limited return of movement if some fibers remain intact. The outcome is determined by the extent of structural preservation. Once complete destruction occurs, recovery is not achievable. Secondary processes such as inflammation and edema may temporarily worsen function in surrounding tissue, but these effects do not reverse the core loss once necrosis has occurred. Reducing swelling may improve conditions in partially affected areas, but it does not restore dead neurons. The distinction between reversible secondary effects and irreversible primary damage is critical. The primary lesion defines the permanent deficit. This irreversibility boundary represents the point where the condition transitions from an acute vascular event into a chronic neurological state. The initial cause may resolve or stabilize, but the consequences persist because the underlying structure has been altered permanently. The system no longer has the components required to perform voluntary motor output. The failure becomes part of the organism's baseline function. The final state is defined by preserved cognition and awareness, combined with permanent loss of voluntary movement, established by the structural destruction of motor pathways within the brainstem. This is not a condition that fluctuates or resolves spontaneously. It is a fixed outcome determined by the extent of neuronal death. The boundary between reversible and irreversible has already been crossed. The system cannot return to its previous state. Misdiagnosis window. The absence of movement creates an immediate diagnostic challenge because external observation suggests a global loss of consciousness rather than a selective motor disconnection. The body is still unresponsive and unable to produce speech or gesture, which are commonly used as primary indicators of awareness in clinical settings. Without visible output, the default interpretation often shifts toward coma or reduced consciousness states. This assumption forms the basis of early misclassification. The condition presents as silence rather than disconnection. In a coma, cortical activity is impaired and awareness is absent, resulting in no purposeful response to external stimuli. In locked-in syndrome, cortical function remains intact and awareness is preserved despite the absence of motor output. The distinction lies entirely in the integrity of consciousness rather than movement. However, because both conditions lack visible response, they can appear identical without targeted examination. The difference is internal and not immediately observable. The vegetative state introduces another layer of diagnostic confusion, as patients may exhibit spontaneous eye-opening and sleep-wake cycles without meaningful awareness. This can resemble the preserved eye function seen in locked-in syndrome, particularly when vertical gaze and blinking remain intact. Without careful assessment of purposeful eye movement, clinicians may misinterpret reflexive or random eye activity as evidence of a vegetative condition. The overlap in external presentation increases the risk of misdiagnosis. Visual behavior must be interpreted precisely. Detection of preserved consciousness depends on identifying intentional eye movements, which serve as the primary remaining channel for voluntary output. Patients may be able to move their eyes vertically or blink in response to commands, establishing a communication system based on coded responses. This requires structured testing, such as asking the patient to look up for yes and down for no. Without this deliberate assessment, the preserved function may go unnoticed. Awareness remains hidden behind immobility. The timing of diagnosis is critical because early misinterpretation can delay appropriate management and communication strategies. During the acute phase following the vascular event, reduced responsiveness may be attributed to sedation, metabolic disturbance, or generalized neurological injury. As a result, the specific pattern of preserved cognition with motor paralysis may not be immediately recognized. This delay prolongs the period in which the patient is conscious, but unable to communicate. The diagnostic window becomes a period of uncertainty. Neurological examination must focus on differentiating between absence of response and absence of awareness. This involves assessing brainstem reflexes, eye movement control, and any reproducible response to commands delivered through preserved pathways. The presence of consistent, intentional eye movement indicates intact cognition despite motor failure. In contrast, inconsistent or reflexive responses suggest impaired awareness. The distinction depends on careful observation and repeated testing. Precision is required. Imaging studies such as MRI can reveal lesions in the ventral pons, providing anatomical confirmation of the condition and supporting clinical findings. The identification of a localized brainstem infarct aligns with the observed pattern of preserved consciousness and motor paralysis. However, imaging alone does not establish awareness. It must be correlated with behavioral evidence of intentional response. Diagnosis requires integration of structural and functional data. Neither is sufficient alone. Communication strategies are developed once awareness is confirmed, using eye movement as the primary interface. Patients may learn to answer questions, select letters, or convey basic needs through structured eye-based systems. This transforms the condition from apparent unresponsiveness into limited but functional communication. The process depends on recognizing preserved cognition early. Without diagnosis, this potential remains inaccessible. The misdiagnosis window represents a period where the condition is present but not yet identified, during which the patient remains conscious without the ability to interact. This phase can persist until targeted neurological assessment is performed. The duration varies depending on clinical context, available resources, and awareness of the condition. Early recognition reduces this window. Delayed recognition prolongs it. The distinction between locked-in syndrome and other disorders of consciousness is critical because it determines both prognosis and management. Coma and vegetative states involve impaired or absent awareness, while locked-in syndrome preserves cognitive function despite motor paralysis. Treatment priorities differ accordingly, focusing on communication and supportive care rather than recovery of consciousness. Accurate classification guides intervention. Misclassification alters care. This diagnostic challenge arises from reliance on observable behavior as a proxy for internal state, which fails in conditions where output pathways are disrupted. The nervous system remains active internally, but its signals cannot reach the external environment. This disconnect reveals the limitation of observation-based assessment. Awareness cannot always be inferred from movement. The system requires deeper evaluation. The misdiagnosis window closes once intentional eye movement is identified and the condition is recognized, allowing the patient to establish a communication channel and confirming preserved cognition. At this point, the external appearance of unresponsiveness is replaced by evidence of awareness. The diagnosis becomes clear. The internal state is finally observable. Network consequence. The failure localized within the ventral pons reveals a broader property of the nervous system, where critical functions depend on narrow anatomical corridors that act as convergence points for large volumes of information. The brain does not distribute all functions evenly across redundant pathways. Instead, it concentrates essential signals into compact regions that allow efficient transmission under normal conditions. This design reduces distance and complexity, but increases vulnerability. When one of these corridors fails, the effect extends far beyond its physical size. The brainstem represents one of the most critical of these convergence zones, integrating motor output, autonomic regulation, and communication between higher and lower neural systems. Within this structure, the ventral pons functions as a bottleneck for voluntary movement, channeling signals from the cortex to the spinal cord and cranial nerve nuclei. The loss of this single region does not impair isolated functions. It removes an entire class of output from the system. The result is a network-level failure originating from a localized lesion. This pattern is not unique to locked-in syndrome, but reflects a general principle seen across multiple neurological conditions. In a brainstem stroke, similar disruptions can affect respiration, consciousness, or autonomic stability, depending on the exact location of the lesion. Each region within the brainstem carries distinct functional responsibilities, and damage produces predictable patterns based on anatomical organization. The system behaves according to structure. Function follows location. A comparable vulnerability exists in conditions such as central pontine myelinolysis, where disruption of myelin within the pons interferes with signal conduction across the same densely packed pathways. Although the mechanism differs from vascular occlusion, the outcome can converge on similar patterns of motor disconnection. This demonstrates that different pathological processes can target the same anatomical bottleneck. The result is determined by location rather than cause. The nervous system relies on continuous signal transmission across multiple hierarchical levels, from cortical planning to peripheral execution. When a single node within this hierarchy fails, upstream systems continue functioning, while downstream systems remain intact but inactive. The disruption occurs in the link between them. This creates a mismatch between preserved capability and lost function. The system is operational but disconnected. This form of network failure highlights the absence of redundancy in critical pathways, particularly those involved in voluntary motor control. Unlike other biological systems that maintain backup routes, these pathways depend on uninterrupted transmission through specific anatomical structures. There is no alternate route for signals to bypass the ventral pons once it is destroyed. The architecture prioritizes efficiency over redundancy. This trade-off defines the severity of the outcome. The preservation of consciousness alongside loss of movement further emphasizes the modular organization of the brain, where different functions are compartmentalized into distinct but interconnected systems. The reticular activating system remains functional, sustaining awareness while motor pathways are eliminated. This separation allows one system to continue while another fails completely. The brain does not shut down globally. It fails in defined segments. This segmentation also explains why the condition remains stable once established. The damaged pathways do not regenerate, and the preserved systems do not compensate for their loss. The network does not reorganize to restore voluntary movement because the required connections no longer exist. The system reaches a new equilibrium defined by structural constraints. The state becomes permanent. The implications extend beyond a single condition, illustrating how neurological disorders often arise from failure at key points within a network rather than diffuse damage across the entire system. A small lesion can produce extensive functional loss if it occurs at a convergence point. The scale of dysfunction reflects the importance of the location, not the size of the lesion. This principle applies across multiple diseases. Localization defines outcome. The brainstem's role as a central relay makes it one of the most critical structures for maintaining integrated function across the nervous system. Damage here does not isolate a single function, but disrupts communication between entire systems. The result is a breakdown of coordination between cognition and action, perception and response, command and execution. The network loses coherence. The system becomes fragmented. The condition demonstrates that normal human function depends not only on the integrity of individual components, but on the continuity of connections between them. Neurons must not only exist, but communicate through intact pathways. When these pathways are interrupted, even intact systems become functionally silent. The failure is not in the components themselves, but in their inability to connect. The network defines function. The final state reflects a system where higher-order processes continue to operate without the ability to influence the external environment, while lower systems remain capable, but inactive due to lack of input. This creates a persistent division between internal activity and external expression. The network remains partially active, but functionally incomplete. The condition is defined by disconnection. This outcome reinforces a fundamental principle of neurobiology. Function emerges from connectivity, and loss of connectivity results in loss of function, regardless of the integrity of individual structures. The ventral pons serves as a critical node within this network, and its failure reveals the dependency of the entire system on a narrow anatomical pathway. The system does not fail gradually. It fails at its points of convergence.
Pathology of Fear is a medical documentary series presenting clinically documented neurologic and pathological mechanisms for educational analysis.